on. We're a bit too new. A bit too new to present. So I decided to present something I care a lot about. And based on now over 10 years of experience doing machine learning in biology, which is model validation when doing machine learning in biology. Okay, I got it. So validation of machine learning models for biological discovery. And the reason I'm interested in this is both as someone who develops methods, machine learning methods, I want to make sure that my methods work and that I can compare what I'm doing with other methods. But also I would say more recently, since I came back to France, I've been working with people who are really interested in discovering new things in biology, which means biologists. And so we were a bit worried about some of the things we're doing in terms of making sure that before we send a list of 1,000 genes to our collaborators to validate that it would make sense. So I will start this presentation with a bit of a machine learning 101 course, just to make sure that we're all on the same page. And then I will talk about I would say old project and how things we've learned in terms of validating how to build a 3D model of DNA from high-C contact maps. And then something a bit more recent, which is more about predicting protein function from its sequence. So it's really a lesson learned kind of presentation. So machine learning, so what is it? So of course, as anyone, I went on Wikipedia to find the definition. And so Wikipedia tells us that it's a field of study of artificial intelligence concerned with the development and study of statistical algorithms that can learn from data and generalize to unseen data. So I think at this point, almost everyone knows what machine learning is, but it's all the algorithms that typically work very well when you have a large corpus of data and too complex to analyze by hand. Right. So broadly, we have supervised learning where our data is labeled. So here we have 2D points with, can I use a pointer? Nope. I have, no, well, here. So we have an unlabeled point and okay, unlabeled point. And the question is, can we find delimitation from the blue class and the red class that will then allow us to label it? So just finding a way to separate the blue points and red points. And then we have unsupervised learning, which is all clustering, dimensionality reduction. So can we find some structure in this data? So to give examples of learning for biology, I'm going to present you very briefly 3 projects that we currently have ongoing in the lab. So the first one is a project about discovering novel secretion system types in bacteria. So there are complexes of protein that bacteria uses to interact with the environment. And they're really hard to characterize in the lab. So currently there's 10 types known. And so the question was, can we build machine learning techniques that use protein sequence and genomic organization that will be able to help us discover novel types of secretion systems? And the idea was a bit that if it takes 10 years in the lab, maybe we can do it a bit faster with machine learning. This is still to be confirmed. Then we have another project, which is, I would say, more or less biological data, which is about biosensor calibration. So we have colleagues here that develop biosensors. So they're small devices that use enzymatic reaction to measure elements. So in this case, it's measuring glucose that we're interested in. And the problem that they're having is it works really well, but over a very short period of time. So the idea was, can we extend the life of those biosensors by building techniques that would help us recalibrate the device? And then the last one is about characterizing the gut microbiome, because we have, again, collaborators from the lab that developed a small device. It's like a capsule about the size of an ibuprofen that you ingest. It opens in your gut. It collects gut microbiome, gut samples, and then you can send the sample to sequencing and get the bacterial composition. And we were interested in seeing where this capsule would open, because this is going to depend a lot on the people who ingest, on the pH in your gut and things like this. So we're currently working on this. So out of these three projects, I would say the first one and the last one are very interested in biological insight. We're interested in discovering new elements, either about the gut microbiome or new elements in terms of the secretion system. And this one is a bit more of a classical machine learning problem where the end goal is just reproduction. Okay, so how do we validate machine learning approaches? If we go back to the definition, there's something interesting here, which is that machine learning needs to generalize data. And making prediction is hard, but it's especially about the future. So how can we validate machine learning techniques when the goal is to predict on unseen data? And just to show, like, give a brief idea of why it's a hard problem, we go back to the supervised learning approach. And if you ask two people to delineate the blue class from the red class, you'll have potentially very different answers. And so the question is, which model works better on, for example, this data set? In this case, the solid line makes no error, but it's not really what we want. We want to know which one is the most likely to be correct. And this is where it becomes a bit tricky, because what you need in order to compare those two models is to estimate what we call the generalization error. So the error you're making on data that you don't have. So very classically in machine learning, the way we do it is we first define a metric, which is a function that takes your prediction and your labels and returns a score. And then the goal is to do a control estimation of this error on data that you don't have, hence the expectation here. The way we do it is now, I think, very well known, is we take our data, we split it into a training set and a test set. A test set that is typically 10% of your data. And of course, there's this whole trade-off between better learning versus better estimation. This test set is meant to be making what we call external validation in some field of research. So in psychology, it's something that is quite well known. So the goal is to have something that is as close as possible to whatever you're going to apply your machine learning algorithm to. So there's a couple of things you need to be a bit careful about. The test set needs to be different enough from the training set. So typically you don't want to have a repeated acquisition of the same individual in your train and test set, because then the problem becomes more simple. And in some cases, you can really mimic what you're trying to apply your machine algorithm to, which is, for example, take data from a new site or later in time, etc. And then the other thing is you need your test sample to be representative of your target population. So, for example, if you're developing a biomarker and you want to apply it to a hospital, and you know that the population in your hospital is maybe a bit different than the hospital has been at, well, you want to make sure that your test works in your setting. So it can be, so when I was working in Berkeley, one was the hospitals we were working with had a lot of people from Spanish origin or Mexican origin, and typically the biomarkers would be done on people who volunteer for studies, so a lot of white population, and it doesn't necessarily generalize to the population we're applying the biomarkers to. Okay, so I've spoken about supervised learning. In terms of unsupervised models, it's a bit more complicated. And we typically go back to, okay, what do we really want from these models? And typically with unsupervised models, we want to make the data more understandable. And this is when the validation strategy becomes very linked to what we want, which is You can have like favoring simple models over complex one. You want your models to be coherent with results. And then another way to validate is just to integrate your model with others of data to make sure that they make sense. So it's a bit more handier. So just in summary, for supervised learning, we have a very well-defined validation strategy. You choose a metric, you split your data in train and test, you estimate the generalization error. If it's good, it should work fine. And then for unsupervised learning, it's a bit more do it as you think it's right, and hopefully it will be okay. Okay, so now I am going to give you what I've learned about validating machine learning models. So it's not necessarily published results in the sense that it was more insights that we gathered along the way. So the first element I'm going to talk about is when I was working on inferring a 3D model of a DNA from a contact map. And really, we had a bit of problems with success metrics. So to give you a brief introduction about what we're trying to do, I was working, so this is projects that are done on high-C data, where we have a contact count metric. So here is an example. In the case of the Plasmodium falciparum, I've only plotted 2 chromosomes. So it's a squared matrix where each row and each column correspond to a genome window. So for example, in this case, it's a 10 kb window. And each entry of a matrix is the number of times we've seen those 2 regions interact with one another. So here, there's a couple of things that you can very quickly notice. Is that there is a strong red diagonal. They correspond to elements that are adjacent to one another in the genome. So of course, they're closing 1D, so they're closing 3D, and as such, they interact a lot. But we can see that there's other kind of structures. So for example, here, we have a hotspot of interaction between chromosome 8 and 7, which corresponds to the centromere in the Plasmodium falciparum. And it's something that was already known before we were starting to work on this, that the centromeres, like in yeast, tend to co-localize in one space in the nucleus. So all of them tend to interact with them. And so what people have been trying to do is, well, we have a contact count matrix, infer a 3D model of the structure of DNA. So this is one of the questions that I am still actually trying to answer. Can we infer a good 3D model of a genome from a high-C contact count? Now, there's been a wide number of approaches. Most of them are based on the same idea that we're going to model chromosome as a series of beads and then try to fit the beads in a 3D space such that it minimizes the cost function. And here I am more interested in talking about, okay, we have different approaches. How can we compare them? This kind of problem is really close to dimensionality reduction in machine learning. So a lot of the techniques are actually just, like, I would say, traditional dimensionality reduction techniques in machine learning. The first approach that people have used, so me and others, is, well, the easiest is to compare to a known structure. The problem with high-C data is that we don't have a known 3D model for different reasons. One of them is the fact that the models that we're inferring are not actually 3D structures. And then the second is, even if it was, how would we measure this 3D structure? So what we've been doing is just simulating a known structure and then simulate data from it, then infer the structure, and then we can just compare X with X true. The small thing that we need to take into account is these structures are typically defined up to a rotation, translation, and rescaling factor. But comparing these 2 structures is something that is, well, we know how to do well because protein structure inference has done it before us. And typically what we use is the root mean squared deviation, which is a good metric, where it's just that our inferred structure needs to be translated, validated, and rescaled. But it's something we know very well how to do. The problem with this is, of course, that the result will depend a lot on your data simulation strategy. So the shift towards, well, how can we use real data in order to compare 3D inference methods? And so in order to do this, it's, again, taking a step back and thinking, what do we really want from our 3D models? And what we want is typically models that can be interpreted in some fashion. And in order for any form of interpretation, there is something that needs to happen. And I'm quoting Yu, which is a statistician. And she said that in order for any scientific discovery, if you want anything that you can interpret, what you need is reproducibility. And by reproducibility here, what she means is statistical reproducibility, which is you want your result to withhold perturbation on your data. Of course, reasonable perturbation on your data. So it can be on data or on models. So the goal is going to be to use real data and perturbate and see if we can do something with it. In this field of 3D model inference, what we've done is use two contact maps that are somehow related, infer two structures, and then compare the structures, obtain a score, and the more stable your method is, the better it should be. So in terms of perturbation, what we've done is use, for example, different biological replicates. Either if you have, I would say, the possibility of having true biological replicates, then of course using this, or changing a bit the protocol. So changing restriction enzymes between the two contact maps, which gives natural perturbation to the data, and then infer the structures and compare them. We've also done this doing different resolution. So for example, comparing structures inferred at 50 kb versus 100 kb. The idea being that if you change resolution, you should still have models that are roughly the same. And then also different coverage, for example, downsampling the contact maps. And the idea is the more stable the method is, the better it is. Okay, so then we need to compare two structures that are now inferred. We can do this using just RMSD, as we did with simulated data. But there's a bit more complexity in using the RMSD in this case. And it comes from the fact that there's two observations we've made. The first one is that RMSD is size dependent. So if you take a structure, you multiply it by 10, then your RMSD is going to be 10 times bigger, roughly. So you need to rescale X1 so that if you're comparing two methods and they give you very different models in terms of scale, that you can actually compare the RMSD. The other element is that it's not symmetric, but this is just an easy fix. You return the average of the RMSD between the two structures. Okay, so we knew that the RMSD was dependent on scale, but at some point, I think not that long ago, about 3-4 years ago, I was like, well, I'm going to test different rescaling strategy. So I'm going to show you the different results. So the first thing I did is I rescaled the structures such that the average adjacent distance were roughly equal to a constant. And then, so I'm showing you the results on human data where we compare different replicates. And I think it's the 100 kb plots that I'm showing you. So each point of each method corresponds to one chromosome, and I've done this with the whole set of chromosomes that I had. And I've run this on all of these methods that I had for the inferred structures one. And there were already a couple of things that I was a bit surprised with. So the first thing we saw is that using this, the MDS, so all of these three methods and this one are MDS-like methods. But the one that worked the best is this one, which I knew for a fact didn't work very well because it was the first method I tested during my PhD, and we moved away from it because it didn't work very well. And yet you're using this, it was giving the most stable results. The other thing that was a bit surprising was actually, I'm going to mention this later. Okay, so I was already a bit surprised. So then what I did is I reskilled the structures such that all the beats fitted in a circle of diameter. So it's a bit like I have a nucleus, everything needs to fit in it. And now the ranking of the methods changed drastically. So MDS, which worked best in my first rescaling strategy, now was the first one. And then SuperRec now moved from being the worst one to the best one. And I was also a bit surprised like this because I knew SuperRec had outliers. And so we didn't do anything to the outliers, so I didn't expect SuperRec to be the best one. So in the end, what we ended up doing is rescaling such that 90% of the beats fit in a circle of diameter R, and we had yet another ranking in our methods. So for me, when I did this, it was 2 weeks prior to submitting the article. I had a bit of a existential crisis where I was like, is everything we've been doing for 7 years totally wrong? But there's a couple of lessons to learn from this. So the first one is understand the particularities of the metric you are using. So I'm showing an example here on 3D structured modeling, but if you do a supervised learning technique, it's exactly the same thing. If you have a very imbalanced problem, don't use accuracy because then 98% accuracy when you have 98% negative labels doesn't mean anything, right? So we really need to think about the particularity of the metric that we are using. In our case, we're also discovering that what I call the topology of a structure had a huge impact on how we, depending on the metric we were using, it was just favoring a type of methods over another type. Use several metrics as well. So this is something we're already doing. So I knew that with our MSD, we had a problem the moment I saw that we had completely different results than using, for example, person correlation over distances. And then the first one, which is obvious, right? But stability isn't everything. We need to make sure that it makes sense in terms of biology. And just Schnegg and Pestis NB have roughly the same RMSD scores, but the structures don't look at all alike. So stability isn't everything. I can make a very stable method that doesn't give any biological insight. I would say it's actually the easiest. Right? You just return random, but fixing the seed should work fine in terms of stability. But yeah, so, and also look at your data and look at your results. Okay. So I'm now going to move on to a second part, which is about predicting a protein function from its sequence. And here it's a much more recent project. And we had, I wouldn't say we had issues, but we put a lot of thoughts into creating a test set. So I'm going to talk about a bit different strategies and the impact on the results. So this is, it's a huge field of research right now, which is, can we predict a function, the function of a protein from its protein sequence? And so, of course, the answer is roughly yes, because this is how we are annotating a lot of the functions by just comparing unknown protein sequences to known proteins that we know the sequences of. But the goal here is to not use sequence similarity, but more machine learning models. And it's something that protein is part of a secretion system. Now, I'm not going to talk about this particular project, but if you're interested, you should go see poster 240. And instead, I'm presenting a more well-studied problem, which is, can we predict whether a protein is an effector or not? So is it being secreted? And the only reason, so this is results I've generated about 3 weeks ago. And the only reason that I was able to generate the results is because we've done all this work for the project on secretion system. So problem to work. There's another problem, which is most proteins are not effectors. So we're in a imbalanced learning problem, which I'm going to ignore in this presentation. But this can cause issues with the machine learning step. And by imbalanced learning, I'm really in the setting that I was mentioning. So probably 98% of the works well in the lab, and we know very well. But it gives us a lot of uncertainty over a large number of organisms that we have data from. And so we know with certain extremely good. So it was okay. So when we think a bit more about our problem, it turns out that we have structure in our data. So in particular, in our annotation step, we're looking at sequence similarity to known effectors. So it turns out that our proteins can be clustered in terms of sequence similarity. And if we want to be a bit closer to our what we want to discover novel effectors families for which we don't have annotation. So it's akin to basically removing either an HMM profile or a known sequence from our annotation strategy. Now, if we do this, what it means is we're just here considering our proteins that match to the yellow class as unlabeled. So we've sort of hidden our gene families because they're not known. When we look at our ROC curves, already it's a bit different. It works very well up to a certain point where we're starting to not learn anything. Using this strategy, it also means that we can see for unknown gene family, what is our true positive rate. And if we do this on our fivefold, what we can see is that there are gene families that are very well reconstructed, very well found, some that are somehow partially found, and then a bunch of them which are just not discovered at all. So already being closer to what we want in the real case, we're degrading the results a lot. And then the performance does change a lot based on our fold. Then as a next step, what we did is, well, turns out that it's even a bit more complicated than this, which is there are gene families that share a common ancestor. So now if we decide that some gene families are linked and thus have more chance of being discovered through, I would say, traditional phylogenomics approach, and we're hiding now not just gene family that are unrelated, we're choosing to hide gene families that are related to each other. So we're considering gene families with common hits as unknown in our training. So in the case of sequestration system, we can go a lot beyond that because we have a good knowledge of the common ancestors between different sequestration system types. And we do our ROC curve again. It doesn't degrade the results as much. And the same thing for true positive rates. It's a bit similar to what we had before, with some folds not working very well, while some folds work a lot better. Okay. So just in terms of lesson learned in this case, different means of performing the cross-validation give very different pictures of success. It's in our setting, we did even something a bit more complicated than this, was we considered also constructing test data from organisms that were very phylogenetically distant. So we took clades and put them in our training data and test data. Now, this result might be a bit disappointing, right? It's there's about 60 new effectors that we're discovering versus 170 that we're not finding at all. But if we think a bit about what we care about, which is discovering novel effectors, in terms of biological insight, it's huge, right? There is suddenly 50 new effectors that don't have any sequence similarity, no common ancestor that we're finding. So it's not as disappointing as what we could think. So I'm going to start the conclusion. So I've showed two projects where the impact of the metric really changed our perception of what method works well. And then I've shown also the impact of how we construct the test set in terms of model performance. Now, in practice, this is very machine learning ways of validating models. And just to showcase the difficulty of this was to showcase the difficulty of validating machine learning models. But in practice, there is a broad number of ways that we can perform validation. So the typical machine learning approach with metric plus test sets. I didn't speak at all about some of the difficulties that we can have over overfitting. So there is a broad amount of things that we still need to be careful about. But one of the things that we do and that we should be doing, even when we do machine learning models, is to confront our knowledge with other source of data. So in the case of our either secretion system inference or the effector, so if you consider that you're predicting effectors, it's well known that in bacteria, toxins tend to be close to antitoxin. So this is additional knowledge that we can integrate in the validation of approach. The other thing that is well known is that toxins or effectors are close to secretion system. So this, again, it can give us a better idea of if we're sending data for validation to make sure that our collaborators are not wasting time with effectors that have less chance of being effectors. There's another element that I think is super important. It's to develop benchmark data for validation. And there's either benchmark data for data challenges, such as DREAM, but there's also data set created uniquely for the purpose of a validation. So to give you an example of such test set is in proteomics, there's a number of data sets where people have been using a protein from an organism such as mouse or yeast, and then spiked it with proteins from another organism such as human, and then at different concentration. And this is to mimic differential abundant proteins in different samples. And then they've run it through the whole proteomics pipeline. And then suddenly you have a means, because you have a ground truth, to look at whether, for example, your missing data imputation works better than others, because you can go back to differential expression analysis and check that it's the spiking proteins that are differential. So the problem with that, it's typically data sets that are only created for purpose of benchmark. They're costly to create. So it's hard to convince people to create such data sets. And then, of course, typically we want to move on to experimental validation. So we have also a couple of projects where the goal is really to develop biomarkers for specific diseases. And in this case, we always go to a new validation set. So typically a new cohort of patients, and often not from the same hospital we did the initial study. Or of course, any form of complementary analysis. So with this, I want to thank my collaborators. So especially the CompBio working group of 3, and Sophie, Renato, and Sandy Frank, who have done, I would say, all the work in terms of extracting, annotating, extracting features, and we spend a lot of time thinking about how can we validate our secretion system, novel secretion system. And then Jean-Philippe and Bill Noble, who have been my collaborators on the 3D infrastructure since the day I started my PhD.